Imagine your brain as this uh, incredibly high-tech, perfectly climate-controlled server room.
Right.
It's running thousands of critical operations every single second. The lights are blinking, the fans are humming. Everything is in this, you know, perfect precarious balance. But then disaster strikes.
The power gets cut.
Exactly. The whole grid goes dark. Now, logic tells you the biggest danger to those servers is the time they spend powered down, right? Like you're worried about the data degrading in the dark.
Sure, that makes sense intuitively.
But what if the real damage, the catastrophic frying of the motherboards, actually happens the exact moment the power gets violently flicked back on?
It's a paradox, honestly, and it makes biological systems incredibly fragile. I mean, the sudden return of energy can be far more destructive than the actual absence of it.
And that paradox is exactly what we are unpacking in today's deep dive. We're looking at a fascinating 2024 paper from the Department of Anatomical Sciences at Shahid Sadoui University.
Yeah, really rigorous study.
Very rigorous. And their mission was to see if an internal backup system, specifically an ancient plant compound, could act as a prophylactic patch to prevent that server room from just completely melting down when the power surge hits.
Right, and to understand that, we have to define what the power outage actually is in this context. We are looking at transient global cerebral ischemia, or GCI.
GCI. So what does that look like clinically?
So to map this to your server room analogy, GCI isn't a focal stroke where, say, a single cable gets cut, starving one specific sector of the brain.
Right.
It is a severe, widespread drop in cerebral blood flow. Think of a cardiac arrest. The heart stops, and the entire brain is abruptly starved of oxygen and glucose.
So the entire grid goes down.
Yeah.
And because our neurons are incredibly energy hungry, I mean, they don't store their own fuel reserves like muscle cells do, right?
Yeah, not at all.
So the starvation clock just starts ticking immediately.
Instantly. Within minutes of that hypoxia, the neuronal membranes start to depolarize. You get this massive, uncontrolled influx of calcium into the cells.
Which is toxic in those amounts.
Highly toxic. And the mitochondria, the power plants of the cell, they become uncoupled. Their normal metabolic pathways just completely break down. But you hinted at the real villain here earlier.
The power surge.
Exactly. Ischemia reperfusion injury.
Right, because if you are lucky enough to be resuscitated, your heart starts beating again, blood rushes back into the brain. You would think oxygen returning is this grand rescue moment.
You would think so.
But instead, that rush of oxygen hits those damaged, uncoupled mitochondria and creates this massive wave of oxidative stress.
Because normally oxygen is the final electron acceptor in the cellular energy chain. But when the chain is broken from the ischemia, those returning oxygen molecules don't get processed correctly.
They go rogue.
Yeah, they basically pick up stray electrons and become reactive oxygen species, or ROS. These are highly unstable, frantic free radicals. They're missing an electron, and to stabilize themselves, they will literally rip electrons from absolutely anything nearby.
So to stick with the server room thing, it's like the power surge blows out the transformers, and now you have live sparks shooting onto the server racks, just this chaotic demolition crew indiscriminately destroying the hardware.
That is a very accurate way to picture it. And when these reactive oxygen species attack the polyunsaturated fatty acids that make up the cell membranes, it kicks off a destructive chain reaction.
Lipid peroxidation.
Exactly. They're literally shredding the cell's outer envelope. They oxidize critical proteins, they mutate DNA, and ultimately this overwhelming cellular damage triggers apoptosis.
That's cell death.
Right. The cell realizes it is too damaged to function and initiates its own programmed death.
And the main casualty of this surge, like the server rack that seems to catch fire the fastest, is the hippocampus, specifically the CA1 region.
The memory center.
Yeah, for anyone listening, if you want to consolidate short-term memory into a long-term memory, like where you left your keys or a conversation you had yesterday, you completely rely on the pyramidal neurons in your CA1.
You do.
Why is this specific region so incredibly vulnerable to that reperfusion injury?
Well, it comes down to something called selective vulnerability. The CA1 pyramidal neurons have an exceptionally high metabolic rate, meaning they demand a lot of energy just at baseline.
Okay, so they're already running hot.
Exactly. And they also have a very high density of specific glutamate receptors. When ischemia hits, there is this massive release of the neurotransmitter glutamate, which overstimulates these receptors.
Which drives even more of that toxic calcium into the cells.
Right. So following a GCI event, patients often experience a delayed selective death of these exact neurons. and that leads to severe post-ischemic cognitive impairment and frequently vascular dementia.
Wow. Which brings us to the intervention the researchers were testing.
Mm.
If our natural antioxidant defenses just get completely overwhelmed by this ROS surge, we need to precondition the brain, right? We need reinforcement.
We need to boost the baseline.
Enter berberine or BBR. Now, this is an isoquinoline alkaloid isolated from traditional Chinese medicinal herbs, like the Coptida species. People have used it for centuries, mainly as an antipyretic for fevers, gastrointestinal issues.
But from a neuropharmacological perspective, berberine has a very specific, very rare advantage.
Oh.
It is highly lipophilic. That lipid solubility, combined with its unique molecular structure, allows it to easily penetrate the blood-brain barrier.
Which is usually the hardest part, right?
Oh, it's the absolute hardest part of designing any neuroprotective agent. You can have a miracle molecule in a petri dish, but if it can't get past the tight junctions of the BBB, it is clinically useless.
Right. But berberine crosses that checkpoint.
It does, easily.
Okay, let's unpack the experiment. So the researchers set up a controlled GCI model using 63 adult male Wistar rats. They divided them into 3 distinct groups.
Standard experimental setup.
Yeah. The sham group, which underwent the anesthesia and surgical neck incision, but without the arterial clamping, so that's our baseline control. Then the untreated GCI group, where the common carotid arteries were clamped shut for 20 solid minutes to induce the ischemia, followed by the reperfusion.
And then the critical third cohort, the GCI plus berberine group. These rats also had the 20-minute occlusion, but they were given berberine via intraperitoneal injection, 50 milligrams per kilogram.
Now, wait, I want to pause on the methodology here, because the timeline of that injection is like the entire crux of the study.
It really is.
They didn't induce the heart attack and then rush in with a berberine syringe, right? They injected the rats with berberine once a day for 7 full days before the simulated ischemia, and then one final dose 6 hours after.
Right?
Are we talking about a treatment here or a daily preventative vitamin? I mean, they preloaded the system for a whole week.
That's a great distinction to make. This is strictly a prophylactic study design. They are not investigating acute stroke rescue protocols here. They are asking a very specific biochemical question. If we saturate the brain's microenvironment with this alkaloid for a week, does it alter the baseline cellular defenses enough to withstand a catastrophic ischemic event?
It's about conditioning the tissue prior to the insult, giving them a head start.
Exactly.
So 24 hours after the reperfusion, they examine the brains. The first thing they look at is the blood-brain barrier itself, because if the perimeter falls, everything else falls. I originally thought of the BBB as like a bouncer at a club, right?
Yeah.
Keeping the bad guys out.
It's a good analogy.
But based on the lipid peroxidation we just talked about, it's more structural than that, isn't it?
Much more structural. The blood-brain barrier is primarily composed of endothelial cells stitched tightly together by specific proteins, claudins and occludins, forming what we call tight junctions.
Okay.
During GCI, the oxidative stress and the inflammation activate enzymes called matrix metalloproteinases. And these enzymes literally digest those tight junction proteins. Yeah. The barrier doesn't just fail, it physically degrades. The tight seams of the blood vessels become porous.
And when it gets porous, neurotoxic molecules, inflammatory cytokines, fluid, it all just leaks directly into the fragile brain tissue.
Right, and to quantify this leakage, the researchers use the Evans blue dye test.
Oh, I read about this. They inject the dye into the rat's tail vein.
Exactly. Now, in the sham rats, the BBB is intact, so the brain tissue stays clear. The dye stays in the blood. But in the untreated GCI rats, the spectrophotometry showed massive concentrations of the blue dye actually inside the brain.
Because the tight junctions were destroyed, the bouncer was knocked out.
The club doors were wide open, but conversely, in the rats pre-treated with berberine, that dye leakage was drastically reduced. The concentration in the brain tissue was vastly lower.
So how is the berberine doing that? Is it acting like a molecular warder for those tight junctions? Giving the bouncer a Kevlar vest?
In a way, yeah. By suppressing the initial wave of oxidative stress we talked about, berberine prevents the overactivation of those matrix metalloproteinases. It stops the enzymes from digesting the claudins and occludins in the first place. So the tight junctions remain intact and the structural integrity of the barrier is preserved.
Okay, so the perimeter holds, the toxic fluid doesn't flood the system, but what about the cells inside that were still subjected to 20 minutes of severe hypoxia? The power still went out.
True.
The researchers used stereological analysis, which, for the listener, is a highly precise way to estimate 3D volumes and cell numbers from 2D microscopic slices. And they look directly at the CA1 region.
And the visual evidence under the microscope is just striking. In the untreated GCI group, stereology revealed severe volume loss across the CA1 and its sublayers.
The Orions, pyramidal, and radiatum layers.
Yes. When you look at the pyramidal neurons themselves, you see the classic morphological hallmarks of apoptosis. The cell bodies are shrunken, the nuclei are darkly stained and fragmented.
They're dead.
They are effectively dead or dying, yes.
But the Berberine group showed a massive structural rescue, right? The volume loss in the sublayers was heavily mitigated. And more importantly, the actual numerical density of healthy pyramidal neurons was profoundly higher than in the untreated rats.
He was night and day.
So Berberine didn't just delay the damage, it actually preserved the physical architecture of the memory center.
It did. But to truly understand the mechanism of that preservation, we have to drop down a level and look at the biochemical assays.
The chemical warfare level.
Exactly. We know berberine preserved the tight junctions and saved the neurons, but we need to look at the endogenous antioxidant pathways to see how it fought off those reactive oxygen species.
Right, because the brain has its own defense system. It relies on specific antioxidant enzymes, superoxide dismutase, or SOD, catalase, CAT, and glutathione peroxidase, GPX.
Those are the big three.
These are the enzymes that normally disarm the free radicals before they can shred the lipid membranes.
Right. Correct. It's essentially an enzymatic relay race. SOD is the first line of defense. It converts the highly toxic superoxide radical into hydrogen peroxide.
Which is still somewhat toxic, right?
Yes, which is why CCAT and GPX take that hydrogen peroxide and convert it into safe, neutral water and oxygen.
Oh, okay.
But during GCI, the ROS production is so massive that it depletes these enzymes. They just can't keep up with the surge.
And the physical evidence of them failing is malondialdehyde, or MDA. This is the primary biomarker of lipid peroxidation. If free radicals are successfully tearing electrons away from the cell membranes, MDA is basically the chemical exhaust fume left behind.
Exactly. It's the smoke from the fire. And as you would anticipate, the untreated GCI rats displayed a massive, significant spike in tissue MDA levels. Their cellular membranes were actively being degraded.
But in the berberine pretreated rats, the MDA levels were radically suppressed.
Yeah.
I mean, the berberine essentially halted the lipid peroxidation.
It did, and this is where the mechanism gets really fascinating. Berberine didn't just float around acting as a sponge for free radicals. It actually reprogrammed the brain's enzymatic relay race.
Wait, really? How?
The researchers found that berberine significantly upregulated the activity of catalase. It pushed the CAT activity almost entirely back to the baseline level seen in the healthy sham rats.
Wow.
It also elevated the activity of GPX and SOD compared to the untreated group, pulling the overall antioxidant capacity back toward homeostasis.
But I noticed a nuance in the data here. Berberine completely rescued the catalase activity, right? Like a highly significant statistical difference. But its impact on GPX and SOD, while elevated and definitely better than doing nothing, wasn't quite as dramatically significant when compared directly to the untreated group.
That's right.
Why does this plant alkaloid seem to favor the catalase pathway over the others?
That is an excellent observation, and it honestly points to a limitation in our current understanding of the molecule's specific pharmacodynamics. Berberine likely doesn't just interact with the enzymes directly, it likely acts upstream at the transcriptional level.
Meaning it changes gene expression.
Yes. It's known to activate pathways like NRF2, which is a transcripting factor that moves into the cell nucleus and turns on the genes responsible for producing these antioxidants.
Oh, that makes sense.
So the differential effect on CT versus SOD might be due to how berberine specifically interacts with the promoter regions of those specific genes, or it might just be related to the timeline of the 7-day pre-treatment. The precise molecular binding kinetics still require further isolation.
I love that. The data clearly shows it works, it saves the tissue, it acts like a biological rust-proofing agent. But the exact genetic signaling pathway it uses to play favorites with catalase is still a mystery for the next paper.
Science rarely answers one question without asking 3 more.
Exactly. Yeah. But, okay, let's pull back from the biochemistry and look at the functional outcome. Because you can have perfectly preserved tight junctions. You can have high catalase and 0 MDA. But if the rat still can't remember anything, the neuroprotection is kind of clinically irrelevant.
Right. It has to translate to behavior.
Did the cellular survival actually save their memory?
To evaluate that, they employed the passive avoidance learning test using a shuttle box apparatus. This is a very standard, highly validated assay for evaluating hippocampal-dependent memory consolidation in rodents.
Right. So for those unfamiliar, the shuttle box is this enclosure divided into two distinct chambers. One is brightly illuminated, and the other is completely dark. And the floor of the dark chamber is an electrified stainless steel grid.
Yes, and you have to remember, rats are nocturnal prey animals, so their innate biological drive when placed in a bright space is to immediately escape into the dark space.
It's just instinct.
Pure instinct. So the researchers conduct an acquisition phase prior to the surgery. The rat is placed in the light chamber. Naturally, it crosses into the dark chamber. But the moment it enters, it receives a mild aversive foot shock.
Ouch. So it retreats to the light.
Right. And over a few trials, the rat learns to suppress its innate instinct. It learns that the dark equals pain. A healthy rat will override millions of years of evolutionary programming and remain in the light chamber for the maximum test duration, which is usually about 120 seconds.
Then comes the retention phase, right?
Yeah.
Which they do 24 hours after the ischemia reperfusion event. They place the rat back in the light room. No shocks are administered this time?
No shocks, exactly.
They're strictly measuring step-through latency, meaning how long it takes the rat to forget the trauma of the training and give in to its instinct to seek the dark.
And the results here perfectly mirrored the stereological data. The untreated GCI rats experienced profound amnesia. Their step-through latency was incredibly short. They practically sprinted into the dark chamber, and they spent the vast majority of the test time residing in the exact area where they had been shocked.
Because their CA1 pyramidal neurons were decimated. The physical hardware that held the memory of the shock was literally destroyed by the oxidative stress. The data was wiped.
Precisely. But the Berberine-treated cohort demonstrated highly significant retention of the memory.
That is incredible.
Their step-through latency was drastically prolonged, and their total time spent in the dark compartment was minimized. So the structural preservation of the hippocampus directly equated to the preservation of complex learned behavior.
It is such an incredible proof of concept. From the blood-brain barrier holding the line, to the catalase neutralizing the free radicals, to the CA1 neurons surviving, down to the rat actually remembering the shock. It's a complete vertical stack of neuroprotection.
It really is.
But, as always, we have to look at the clinical reality. Let's be real for a second. This is a rat model. Humans are obviously far more complex. What are the translational hurdles between this 2024 study and, say, a human patient at risk for cardiac arrest?
The primary hurdle is pharmacokinetics, specifically bioavailability.
Right.
The researchers utilized an intraperitoneal injection administering the berberine directly into the abdominal cavity. They did this because berberine has notoriously poor oral bioavailability.
So if I just take a Berberine capsule from the health food store,
It undergoes extensive first-pass metabolism in the liver, and whatever is left is aggressively pumped back out into the gut by efflux transporters like P-glycoprotein.
right, the GI tract basically treats it as a xenobiotic and just gets rid of it before it can ever reach the bloodstream in therapeutic concentrations.
Exactly. So we cannot linearly extrapolate a 50 milligram per kilogram IP dose in a rodent to an oral supplement in a human. To achieve this level of tissue saturation in a human brain, pharmacologists will need to develop advanced delivery systems.
Like liposomes.
Yes, perhaps liposomal formulations, nanoparticle encapsulation, or specific intravenous protocols that can entirely bypass the gut and the liver.
And we also have to consider the prophylactic nature of this study, right? Like we said earlier, this isn't an acute rescue drug you give in the ambulance after the power goes out. The tissue has to be preloaded.
Which defines its potential clinical application. The researchers are targeting this as a preconditioning agent.
Okay, so who gets it?
Well, the ideal use case would be identifying a patient population with severe cardiovascular disease who are at a statistically high imminent risk for an ischemic event, and prescribing a bioavailable berberine therapeutic to proactively armor their neuronal microenvironment.
It is a total paradigm shift. Instead of waiting for the cardiac arrest and hoping the reperfusion doesn't cause dementia, we structurally sortify the tight junctions and upregulate the antioxidant genetics before the event ever even happens.
It shifts the focus from damage control to damage prevention at a fundamental cellular level.
So we've covered a massive amount of ground today. We looked at how a sudden loss and subsequent rush of oxygen triggers a catastrophic wave of free radicals, destroying the blood-brain barrier and shredding the memory-critical neurons of the CA1.
Right.
But by preconditioning the system with berberine, researchers observed a trifecta of protection. It preserved the tight junctions of the BBB, it aggressively upregulated the catalase pathway to halt lipid peroxidation, and it functionally saved the architecture of the hippocampus, preserving the subject's memory.
It is a highly compelling demonstration of how molecular pharmacology can intersect with traditional medicine to address one of the most complex challenges in neurology.
It really is. The fact that an isoquinoline alkaloid, utilized for generations just to soothe fevers, happens to perfectly interface with the specific genetic pathways required to protect our highest-order memories from oxidative starvation is fascinating.
True.
It makes you wonder, if this single ancient compound holds the precise biochemical key to neuroprotection, what other intricate cellular defenses are currently hiding in the natural world?